as said, today I'm gonna tell you a bit about some kind of small overview about the techniques that we do at the Institute of Cell Biology. And since we are working with the urinary bladder, that would be the topic of the talk. So if you will have any questions, you can just raise them. You can stop me during the talk or afterwards, or just contact me and we can discuss or collaborate further on later. So first, some backgrounds of where I'm coming from. Institute for Cell Biology is part of the Faculty of Medicine, University of Ljubljana. I'm actually not the full-time researcher. We are heavily involved in teaching. But on the other hand, the microscopy is, let's say, long-term present at the Institute. We have a rather long history of electron microscopy, and as has to be mentioned, we are also part of Euro-BioImaging. Slovenian node is called Symbion, and so we have some access to other equipments also through this channel. What we are doing is, of course, urinary bladder. More precisely, it's urothelium, so epithelial tissue of the urinary bladder. And our group is actually exploring different aspects of it. Very much we are interested how urothelial differentiation takes place. Being so in physiological conditions, so how membranes are organized, how membrane trafficking takes place, and so on, in order to obtain just functional urothelium, which has this permeability barrier function. On the other hand, we are also interested in what happens during some pathologies. So how some effects of external or internal factors contribute to the diseases like cancer or cystitis. And as a third pillar is actually also some therapeutic approaches, how we try to cure those diseases in order to obtain back normal function of the urothelium. So, and in this talk, I will, let's say, go over 4 parts of it. First, introduction, what is actually more in detail urinary bladder and its epithelium. Then I will show you a little bit about volume EM methods that we are using and what we do with that part. And third part is about our attempts to process those data that we obtain by volume EM in an automatic manner, and then just wrap up, and that will be. So different parts of the talk will be color-coded so that you will be more, that you could follow the talk easier. So urothelium, it's actually the multi-layered epithelium, and it's the tightest epithelium in our bodies, and even broader in the whole mammalian world. Why we are studying this? Because there is quite a need, actually. So, for instance, diseases that affect people are very common. For instance, urine infections. By statistic, 30% of women will actually have urinary bladder urine infection during their lifetime. And if you had it, then you probably know it's not very pleasant thing to have. It's painful. On the other hand, urinary cancer, it's actually rather not the deadliest cancer, but it's perceived as the most expensive cancer to cure. It's very much recurrent, and patient has to be monitored over the longer periods, and that add costs to the health system. So we, as a basic institute, we are studying the normal process during this bladder, then diseases, and all this in order to find better strategies to cure the diseases. And for studying, we are using different model systems, in particular in vitro systems. So we have rather nicely equipped cell labs, but very much we are also happy that we have access to animals. So we have the animal facility here, and this more complex system gives you different level of the data. And of course, then connections with clinicians enable us to test some things with the patients or obtain the samples of the diseased samples from them. And of course, anyone who ever looked at the cell more into detail in, let's say, in the microscope, knows that cells are not so simple as it's frequently proposed in the textbooks and so. First of all, cells are three-dimensional objects and very dynamic, though they contain similar organelles. They are differently arranged, and for in order to understand them, how these processes take place, how they function, you actually have to determine these special relationships among those organelles and cellular compartments. And this holds true for normal, healthy individuals or cells, and also in the pathological conditions. And so let's introduce the urothelium. So here we have the urinary bladder, and its epithelium, this internal layer, is a three-layered epithelium tissue. So we have the basal cell layer, intermediate, and terminally differentiated cells on the luminal side, which are called umbrella cells. So, and those umbrella cells, they are actually characterized by special transmembrane proteins called uroplakins. Those uroplakins, you can see them here in green. This is umbrella cell. And here below, it's are intermediate cells and basal cells. And here is the connective tissue. So those uroplakins assemble quite well-organized structures called urothelial plaques. And those plaques actually line the apical surface of the umbrella cell and also form fusiform vesicles inside the cytoplasm of umbrella cells. So those are those green dots that you can see here. And urothelial plaques and tight junctions, those two structures are the crucial structures that present blood-urine permeability barrier of those umbrella cells and as that of urinary bladder. If we want to see those fusiform vesicles, we have to go to the level of transmission electron microscopy. And fusiform vesicles are very much flattened vesicles. You can find them individually or in a stacks, which quite resemble Golgi apparatus, right? And because they are quite similar in a shape, there were hypotheses that fusiform vesicles actually are formed directly out of the Golgi. So, for instance, one of our questions was how urothelial plaques are formed. And of course, to study this, you need transmission electron microscopes or microscopes to study ultrastructure. And this is what we have. And very early on, we entered this problem. So in a conventional TM, you lose one dimension, right? You always look to the objects as a 2D projections. And if you combine this with some other problems like random orientation of the sample, so being that the same structure, if you look at it at the different angles, can give you a much different picture. For instance, here, these are the microvilli on the enterocytes. But if you cut those microvilli like that, you see a picture like this. So it's very much different. And the third aspect is, of course, the sectioning, which gives you just small portion of the cell. And for that, what you see on the picture as a sphere, for instance, could be a tube in reality. And so for that, you need something that will give you more three-dimensional reconstruction or view of your sample. And so for that reason, we said, okay, we have to move to volume electron microscopy. And this term, volume electron microscopy, or shorter VM, refers to a group of imaging techniques that can be done by electron microscopes and enable 3D structures or reconstructions in nanometric resolutions. That's quite simple. To place volume EM among other imaging methods, we see that electron microscopy has the best spatial resolution. Of course, the consequence of this is that you cannot reconstruct so large objects as with some other techniques. Or for instance, you cannot track dynamic processes, but it gives you good resolution. So it's quite important that at certain point you just correlate the different imaging techniques to give you the full answer. Among those EM methods, there are several, like serial sectioning, electron tomography, serial block phase, focused ion beam, array tomography. The later three of them are probably the most known in today's labs. How those methods are performed. They all start with fixation of the material. So is it chemical or cryofixation? Then you have some dehydration, embedding, and then you have to do the sectioning. The sectioning can be done in two ways. In certain methods, the sectioning is done by ultramicrotomes outside the microscope. For instance, for serial sectioning TM, for electron tomography, or for array tomography. With serial block phase, SEM, or focused ion beam, you do the sectioning inside the microscope. And that can be by ultramicrotome integrated within the electron microscope, or you can do the sectioning by focused ion beam to evaporate layers of material and then expose deeper layers to the SEM column. In all cases, you eventually get a stack of images which differ along the z-axis, and then you analyze those data, you correlate data, you segment the data, and based on those data sets, you can then do the 3D analysis and reconstructions, modeling, and of course, you can also obtain some quantitative data to give you some numbers about the structures that you have in the volume. Which of the methods that you should use is very much based on the questions that you have. There are many papers describing those methods, and they're actually all has some advantages or disadvantages. Most of them now can be automated to a certain degree, but the major thing that you should keep eye on is the dimensions that you can reconstruct by each of those techniques, which also takes some time to do the method. So maybe this slide will show you that more in a nicer perspective. So electron tomography is used for most precise work when you have smaller volumes, but good resolution. Voxel size is the smallest with electron tomography. Focused ion beam methods give you some intermediate voxel size, but you can actually also have the larger volumes of material of your sample to be analyzed in a reasonable time. And then you have the methods where the mechanical sectioning takes place. That means array tomography or serial block phase. And those methods actually give you a slightly lower resolution, but you can analyze larger volumes. So depends on your question, you choose your method. Of course, serial sectioning by TM, it's the first and the easiest, the cheapest method that you can do. For that, it started already quite long time ago. They were using it to study nervous system, and because we are doing the analysis by TM, the resolution is still the best in at least in the X, Y dimensions. Most of the labs that have electron microscopes has also ultramicrotomes, and you just section your sample, you collect the resins on the grid, and then you do the imaging in the TM, and then you manually align the samples and so on. But so actually this approach is relatively actually it's too labor intensive that can be done in a significant manner today. When I was searching about those studies, I've then later found, let's say, citations like this, that those studies were heroic, that took several years to work, and so very labor intensive. And for instance, in these studies from 86, only 300 neurons were reconstructed by this approach. But to put it more into perspective, that's only about 1% of the single mouse cortical column. So actually, if you want to reconstruct the whole brain by this, you could spend decades. So it's not an option. But electron tomography, it's already much better. So it's also a method based on TM. It's very nice to get high resolution, but smaller volumes. The principle of electron tomography is, of course, that you have this semi-thick section, and then you do the tilting of this section under the electron beam. You obtain a series of images, projections, which can be then aligned and back projected in order to reconstruct the data. So you receive the tomogram and then you can do the 3D reconstructions. We first employed this technique in order to actually see how Golgi is organized. Is actually trans-Golgi network connected to the fusiform vesicles? Is there any morphological data supporting the hypothesis that we have direct maturation of Golgi cisternae into the fusiform vesicles? And based on those it's not, just give me a second. And based on those reconstructions, we did then the modeling of the Golgi, of the trans-Golgi network, and all the compartments that lay after the trans-Golgi up to the fusiform vesicles. And what we've seen is that it's not the case that the fusiform vesicles mature just by detaching Golgi cisternae. We have individual compartments leaving trans-Golgi network, and those smaller vesicles fuse together to actually form bigger structures, immature and then mature fusiform vesicles. So electron tomography in combination with some other methods, that's for instance Tokuyasu immunolabeling, or for instance freeze fracturing, or some other histochemical stainings, gave us support or proof that urothelial plaques are formed in the post-Golgi compartments sequentially. So we then, based on this, proposed a new model how fusiform vesicles are maturing. So by sequential organization of individual smaller vesicles into a larger flattened fusiform vesicles. We also characterized those fusiform vesicles. They are really like pancake flattened vesicles which contain urothelial plaque in the middle. This is this bluish color, and we can see here here are organized urothelial proteins, uroplakins, which can be also seen here in the freeze fracturing images obtained by freeze fracturing as smaller 60 nanometer large particles. So electron tomography gave us nice results for when we needed higher resolution but smaller volumes to reconstruct. But what's something bigger? For instance, mitochondria. For that, we need larger volumes. And we did, by other methods, some studies, and we found that mitochondria are very much different in different types of cells, urothelial cells. In low differentiated urothelial cells, mitochondria are like fragmented, you have smaller dots, and then when you go further towards highly or terminally differentiated cells, you have mitochondria as a network, right? So we correlated this process with some markers of urothelial differentiation, for instance, uroplakins, or Golgi, which is also differentiation dependent, has also differentiation dependent distribution. We did some quantifications, and others. So at the end, we came to the question that those differences that we observe within different types of urothelial cells are probably in certain degree the consequence of mitochondrial dynamics. So we have constant fissions, we have fusions of mitochondria, and we can see a little bit of this in the statical images. But of course, if you want to analyze more, we have to go to the volume EM. And for that, we said, let's go with the serial block phase, SEM, as it enables us to analyze the largest volumes. So serial block phase, SEM, it's a method that has ultra microtome within the microscope. Here's the sample, here's the knife. So the sample is sectioned within the instrument, and then you obtain the images by the SEM column. So this method gives you the quite big overall image, and it started for the biological materials in let's say after the year 2000, 2004, let's say. And when we did the first round of imaging, we found that our samples are not prepared accordingly. We seen much of the charging, the contrast was low, the sectioning was not as we would like to, the tissue was too soft. So we had to go back to the lab and start to optimize the protocols. And of course, we usually use cryofixation, which gives you much nicer ultrastructure in comparison to chemical fixation, where the samples are, or ultrastructure is frequently blown up or just not to the level as you can expect with the cryofixation and free substitution. But then we had to increase the contrast. So we introduced more osmium into the sample. That also helped to reduce the charging of the material, gave better contrast. And then we tested several embedding media like EPON, different formulations of EPON, Durcopen, and so. And then at the end, we've seen the success, and we succeeded to analyze the sample. So here's one of the examples what we obtained. So in this case, you can see this is the lumen of the urinary bladder, and you have this superficial cell called umbrella cell being very large, but you also see the intermediate cells, right? And basal cell layer. So you have the complete urothelium that you can observe. And then when you zoom in, you see, let's say, mitochondria being quite dense in the umbrella cell. But if you go down to the, let's say, intermediate or basal cell, the cytoplasm, it's much more empty. And then we did some reconstructions. This is still work in progress, but we actually find that there are many interesting features within mitochondria in those cells, like also branchings, narrowing, and other aspects that could be of biological significance, or let's say contact sites between ER and mitochondria. So the third method of volume EM that we used is focused ion beam. Probably you mostly heard about this method. It's very much getting place in the life sciences also, but it all began with material sciences because those semiconductor manufacturing can actually very much benefit of milling some parts of the material or depositing materials on the any surfaces actually. So today, what focused ion beam in life sciences started in, let's say, 90s, late 90s, and now it's used for all kinds of 3D reconstructions and very frequent also for TM lamellas lift out preparation. In the whole talk, I'm not referring to any cryo VM techniques, but you probably know that most of those techniques, especially focused ion beam, can be done also under cryo conditions. What you do, you take focused ion beam. There are different types of beam that you can have, and then you use this beam to evaporate the material, and then you can use the SEM column in order to visualize the exposed surface, and then you sequentially cut away a layer and then put another series of imaging to reconstruct the whole volume and then do the reconstruction. How that looks like, this is the trench that you mill with the FIP, and then you decide which part of the section you would like, of your sample you would like to reconstruct. Here's the semi-thin sections. We said, okay, let's put this part to be reconstruct, and this is the image in the SEM in the FIP machine, and this part is the epithelium. So we protected this surface area by some depositing platinum on it. Then we cut the trench, and this now black part is actually the place where you can do the imaging. This is the raw image of the larger part, but the final reconstruction looks like this. So here you see a movie of one part of the cell, an umbrella cell, so this terminally differentiated superficial urothelial cell. It's actually larger than this clip. You don't have the nucleus here, but here on the top, it's the luminal side. Here on the bottom, you see the intermediate cells. And within the cytoplasm of umbrella cell, you see those black structures, which are mostly mitochondria and lysosomes. And then you have those rods, which are fusiform vesicles. Then you have some Golgi here and other organelles. So here we have 1056 serial sections, and you see that the cell is very much packed with different organelles. If I skip further, then we can start to talk about segmentation. So if you want to analyze how, for instance, this Golgi is connected to other structures, how it relates, you have to do the segmentation. And to do this, for all the organelles and compartments that you see in those images in, let's say, 1000 sections, if you want to do this manually, it's just no go. It's too time demanding, and it's not really good one. So you have to consider when you're doing volume EM that you have to spend quite a lot of time to find the proper sample preparation method that will actually work for you. Then you do the actual imaging, which can take also several hours or days. And then you have the data analysis descent, which is actually, I would say, the heaviest or the most demanding in time in the proportions of time. So if you want to analyze this, you have to segment each of those structures manually or in automatic way, and then do the reconstructions. So segmentation is a bottleneck. And that was several years ago. We said, let's find a tool, a software that will be able to reconstruct our cell automatically. And at that time, we couldn't find nothing really. That was like 6-7 years ago. There were many approaches in other techniques, mostly to reconstruct some pathologies or medical images, but to segment ultrastructural compartments of cell, that was not the case. So at that time, we said, then let's do it by our own, and we contacted a group in the Faculty of Computer Sciences in the University of Ljubljana, and they took the challenge, and together we developed a new algorithms for segmentation of fusiform vesicles and other organelles within the eukaryotic cells. How the work went, we took this FIPSAM volume as a starting point. We divided the volume to the sub-volumes. Some sub-volumes were manually annotated to be a learning base for the algorithms, and then we segmented it and tried to find the proper pipelines to segment Golgi, endolysosomes, fusiform vesicles, mitochondria, and others. So there were some problems, of course, that we have to solve first. For instance, artifacts of imaging, curtaining, uneven contrast, low contrast, and so on. But that was solved quite nicely. Then there were proposed some pipelines to do the segmentation. And when we compared our algorithms with common algorithms that were on the market at that time, we seen that our algorithms were quite successful. And so we published all this and in several papers. And we also published the whole data set of urothelial cell that is available. You can find it and use it. It's called UroCell, and be welcome to check it. So to our knowledge, that data set is the first FIPSAM data set that was not obtained from the brain tissue, that has multiple organelles being segmented and annotated, and actually presents quite diverse cell in comparison to other data sets. Lots of those publicly available data sets actually are done on the cell lines or the simple cells from the cultures, while our data set is based on the actually fully mature cell from animals. So what we are doing now, we are trying to optimize some and upgrade some algorithms to be more prone to some artifacts and errors. And particularly, we are interested to improve the algorithms that they could segment better some structures which are hard to divide, to separate, like fusiform vesicles in the stacks, or some structures which can have significance in biological terms, like branching of mitochondria, narrowing of mitochondria, and so on. So the results that we have are, for instance, here. This is the one reconstructed volume of FIPSEM obtained data, and this is then the basis that you can do quantification of your data, how many fusiforms you have in such cells, what happens with the mitochondria, do they branch, do they contact, and so on. And here I would like to just wrap up the whole thing. So in general, urinary bladder epithelium, I explained a little bit how it works, how it forms the permeability barrier, but to be more broader, by analyzing distribution of organelles and compartments within cells, actually you can, that can help you very much to determine the function of those compartments, and with that the whole cell. For the VM methods, there are several options. Currently, no best volume EM imaging method exists. It's very much based on your question, your needs, and of course, each of the techniques has some pluses and minuses, so you have to consider this. And very much to be stressed is that if you don't have the good sample preparation, then you will have also very weak or bad or misleading results. So garbage in, garbage out rule is also applicable here. And with the analysis of volumetric data, I think that this part is actually the crucial at this period. It's a bottleneck to analyze all those things, and we still don't have the algorithms for automatic segmentations that would be capable of really robust reconstructions of all the data that we can produce now. So this part will develop in the future even more, and it will actually very much influence how we see the electron microscopy today. What we're gonna do in the future, and in general, how we see the future of the volume EM and other techniques. I think it will increase the throughput. The machines are more and more available and more user-friendly. We try to correlate different aspects of imaging to have multi-model reconstructions and analysis to give you to integrate more data together. Cryo VM is also something to be considered, and of course putting all this together. But for sure, if we don't improve this automatic segmentation and post-acquisition processing, all those things will not be fully utilized. So as you probably also aware, volume EM and deep learning protocols are very much in the center of development now. For instance, volume EM was one of the technologies that was also proposed by Nature as a technology to watch on. And with that, I would like to also promote the Volume EM community. If you're interested in this, please visit the website of Volume EM, contact people. There are many resources now available to show you this in an easier way. So very last thing, just acknowledgements. I would like to acknowledge my group, or the group to which I belong. Peter Veranic is the head of the group. Most of the work that I've shown you today was done by me and Professor Okromich, and of course collaborators, Margaret Pavelka and the lab in the Vienna. She actually passed away a few years ago now. Then the group in York University, Tung Tien San and Alice Liang. Then Bruno Humble and the group of computer sciences in University of Ljubljana in Slovenia. And thank you also to Euro-BioImaging and to you all for listening the speech. Yeah.